Hello everyone, my name is Banu and I'll be explaining you about my project on integrative bioinformatic analysis to identify microRNA based biomarkers and therapeutics for the treatment of glioblastoma. The main problem statement of my project is that glioblastoma is a highly aggressive type of cancer which does not have proper treatments or diagnostic methods. So I wanted to find a reliable way to diagnose this type of cancer using biomarkers. And maybe these biomarkers could be used as a therapeutic method. The importance of my project is that I want to identify potential biomarkers for the treatment as well as the diagnosis of glioblastoma to improve patient outcome and even facilitate personalized therapy. This study's findings have showed that the potential of using antisense sequences for antisense therapy is a viable option to treat glioblastoma. The novelty of my project is that I have identified novel microRNA-based biomarkers for glioblastoma. I have designed antisense sequences using in silico tools, and I have evaluated the sequences using different types of parameters. This is the overall methodology of my project. I started with data acquisition, followed by identification of differential expression of genes, then finding out the target genes of the microRNAs that I found before. Then I need to find out what these genes do after target identification via functional and pathway analysis. This is followed by network analysis to find out how these genes are interlinked with each other. After that, I have actually conducted, we have used specified tools to construct the antisense sequences. Then I've checked their specificity and their stability by evaluating their parameters. And finally, I've conducted survival analysis to check with the clinical data that's already present to find out if my biomarkers are actually biomarkers or not. This is the result of my differential gene analysis, where I have used two types of data sets, GSC 90603 and GSC 65626. Both are from the Gene Expression Omnibus. On the left side, you would see the microRNA names and the respective log FC values and P values. P values are essentially essentially indicate the error. The lesser it is, the more accurate they are. And log FC values are calculated based on their expression in cancerous cells compared to normal cells. This is just a continuation of the number of microRNAs I've taken. In total, there are 23 of them. Now, I need to find out which genes these 23 upregulated microRNAs were targeting. And they came out to be these target genes. And to find out how these target genes are interlinked with each other, I have performed functional and pathway analysis via a tool. And I have found out that these are the biological processes. As you can see, they're responsible for protein complex. They're present in the membrane, cell surface, extracellular region part, etc., etc. And when it comes to their cellular components, they're responsible for ion binding, protein binding, organic cyclic binding. I have to mention that all these cellular component and biological processes are actually directly linked to glioblastoma. As you would see in the results I've gotten from the pathway analysis, as the genes highlighted in red are the same genes I've gotten in my studies. And as you can see, both the de novo pathway and the secondary pathway for glioblastoma are being affected by the microRNAs that I have obtained. This is the network, the protein interaction network that I've gotten by using the tool Cytoscape. And from this network, I have taken the top 10 hub genes, which are essentially the top 10 most interacting genes, most influential genes rather. And so to properly decide which of them are biomarkers, I've only taken the microRNAs which are only targeting these genes, which came out to be 16 microRNAs. Then I have used the specific tool called PFRED to construct these antisense sequences for the 16 microRNAs, and also found out their specificity with each other. This can be found out by using a tool that is present in the Vienna RNA server, which has given me the MFE values and delta G values. As a rule of thumb, according to the tool's manual, if the values are lesser than or closer to minus 37 kilocalories per mole, the two sequences are very specific to each other. Delta G essentially determines its stability with each other. So this is for the first 8 microRNAs and their corresponding antisense sequences. This is the continuation of that. Then I wanted to find out the stability of the antisense sequences alone. And this tool, specifically called NewPack, also gives me the stability as well as its secondary structure to find out how it would actually bind with its target microRNA. And these are what I've obtained. As you can see, most of these structures have crosslinks, which indicates that they're highly stable. But one interesting fact is that the antisense sequences for the last 2 microRNA, namely 6295P and 1290, are linear. This essentially indicates that they by themselves are not that stable, but they are actually more specific to the target microRNA. After that, to finally determine if the 16 microRNAs that I have are actually correlated with the clinical data that's present in TCGA, for example, I have performed survival analysis, which gives me Kaplan-Meier curves, where the y-axis is proportion of survival, and the x-axis is day survival. To put it in simple terms, the blue line represents the fact that if this microRNA expression is less, and the red line represents the expression of this microRNA more. So if the red line is below the blue line, that means it's highly risky and the survival of the patient does decrease. And for most of my microRNAs, they do decrease. So this indicates that my microRNAs are actually biomarkers for glioblastoma. However, one interesting fact is that microRNA 1953P, 199A5P, and 1290 have proved that they're not exactly linked with glioblastoma whatsoever. So that means that out of 16, only 13 have actual direct relation to the clinical data that's present. So the updated final list of biomarkers is the following. I would like to, these are my conclusions, and they are: the study identified a list of upregulated microRNAs as potential biomarkers for glioblastoma. The functional and network analysis proved that they are associated with glioblastoma. With survival analysis, I was able to find out the final list of microRNAs, of microRNA-based biomarkers. And the antisense sequences that I have constructed were actually stable and specific, and can be used for antisense therapy. For my project, I would like to acknowledge my mentor, Dr. V. Arna ma'am, Dr. B. Sumitra ma'am, Dr. Nagarnath sir for their support, and the head of our department, Dr. Ashutosh Pandey. Thank you for listening. See you.